Okay, then we'll make a bit of a start. We're right on 3 o'clock, so we need to cover the requirements for Prac 9 for food safety. So for food safety, the plan was to use a special lotion, this lotion called the Glow Germ. So I'm not allowed to put bacteria on your hands, but what we used to do is just simply put some of this lotion, which is actually fluorescent under UV light. So the solution is used to simulate the bacteria on your hands. So what we do is put a little bit of this lotion on your hands, or if you like, a little bit of bacteria on your hands, and then go through various different treatments. Some just give your hands a quick rinse, and some doing a proper hand wash for a full 20 seconds, and then have a look at what happens to the fluorescent lotion, and see whether we're able to actually wash away the lotion and hence wash away the bacteria. Now, I haven't got all this stuff at home, and I'm assuming you guys don't have all this stuff at home. So what I've found is a YouTube video which does a very, very similar experiment to what we would have done in class. So in their experiment, they did something similar to what we had as our treatment one, whereas they simply just, well, the before part is you add some of the germ lotion to your hands, and then your hands become very, very fluorescent, as you can see in the photo there. And then if you had, obviously the more effective the hand washing procedure is, the more bacteria you're actually able to remove. So you can see here, this is very ineffective. There's hardly any fluorescent lotion removed, or hence very little bacteria removed. And when you've got a full 10, 20 second hand wash, you're removing significant amounts of the lotion, a significant amount of the bacteria. All right, so the treatment one, what was done there was after you contaminated your hands with the lotion, or bacteria if you like, all we did was just simply rinse your hands with some water, no washing. And in that video, when you get a chance to have a look at it, you'll see that if we simply just rinse our hands with water and no soap and no scrubbing, very little of the fluorescent dye comes off, or very little of the bacteria comes off. So our result for treatment one, we would say that there's 0% reduction. So there's 0% reduction in the amount of lotion, or 0 amount in the amount of bacteria. Yes, someone's got a question?
We have a different table in our logbooks. Do you want us to apply these results to that table, or complete?
Yeah, I've actually put this up onto Canvas. If you go to week, I think it's week 13, it'll be there. I'll see if I can pull it up so you can have a look.
Yeah, I've got it to download. I'm just wondering how we translate it into our logbooks, which look different.
Well, you've got the Word document that I've provided. Aren't they both Word documents? So you should be able to just cut and paste.
No, just put the results part of the document into the table.
Is the logbook available as a PDF, or is it available as a Word document?
Well, we've printed and bound our logbooks.
Oh, you've already bound it. Well, you can always just paste it over the top. That's fine.
Also,
well, you're going to scan it anyway.
Another question.
Yeah.
With the table that you've provided for volunteer 2, they're missing that. So we just not write results for that volunteer?
No, I've got no results for it. So in the YouTube video, they don't do it the same way that I was going to do it. So that's not available. So I'm just going to cross that out. So when we do it in class, we do cold water and hot water. On the video, they just done cold water. So I've just crossed that out. We've got no results.
For which table?
This table here, treatment 2 is not available. Because on this video, all they're showing is something similar to what we would have done, treatment 1, which is simply rinsing with water And on the video, they go through and do hand washing for 5 seconds, 10 seconds, and 20 seconds, which is similar to what we were proposing. I've actually changed it, the wording actually for the treatment, as you see in red. So it's a little bit different from our original Prac. But it does give us similar results to what we were expecting. So you make those slight alterations to your logbook, or if you want, just copy and paste this new version of Prac 9 over the top onto your logbook. Anything else before we go on? Does that make sense, what happened here in treatment 1? Again, it's easier to see on the video when you look at the YouTube video, but I think it's pretty clear here as well. Maybe I'll make that even a bit larger. So no significant reduction in the amount of fluorescence. So what that tells us is that if you're just simply rinsing your hands with water, it won't remove this fluorescent lotion, and hence it won't remove any bacteria that might be on your hands. Treatment 3, they washed for 5 seconds with some soap and water, and they did get a reduction, but if you look at the before and after, there's still a lot of fluorescence, even after. So I'm just saying, look, it's about a 25% reduction. When they did it for 10 seconds, there's more reduction. There's still a fair bit of fluorescent. So I'm guessing it's about 50% reduction. And then when they did the hand washing for a full, they did it for 20 seconds. We need to update here. When we do it in our Prac, I was asking students to wash for 2 minutes. This is actually shown for 20 seconds, so that's not quite right. Anyway, after 20 seconds, they got significant reduction. That's before fluorescent lotion, after fluorescent lotion. So we removed most of the lotion, which means we'd actually remove most of the bacteria if you were to do a correct hand wash for a full 20 seconds. The next demonstration was to see what happens when we allow cross-contamination to occur. Has anyone ever worked in one of the fast food chains, whether it's McDonald's, Hungry Jack's, KFC, whatever it might be, or in a restaurant? Yeah. So you might have actually undergone some training about cross-contamination in terms of hygiene. Yeah. So you want to tell us about it? Is it hard to tell us about it? You haven't got audio?
I work as a cook, so
Yeah.
there's usually like raw cook and cooked cook, if that makes sense.
Yeah, it
So
does.
raw cook deals with flour and all that
Yeah.
stuff. So I'm allowed to touch the cooker, like
Yes.
put the chicken in the cooker with all the flour and stuff, but I have to
Yeah.
wipe it.
Yeah.
And the other cook is meant to deal with the cooked stuff.
All right. Has that workplace got color coding? So you've got color coded knives, color coded chopping boards.
Yeah. So we got color coded wipes. So green ones are for cooks,
Yep.
stuff. So
Yep.
that includes just chicken, basically cooked chicken, and the red one is for raw stuff.
All right. So you got the chopping boards and the knives color coded, or just the chopping boards?
No chopping boards, and we got like sauces and stuff, but
Oh yeah, okay. But you've got a system in place to make sure
yeah.
there's no cross contamination from the raw product, in your case being the chicken, to the finished product, which is sounds like it's the cooked chicken. I've seen color coding even of people. I've been to infant powder factory, a baby powder factory over in northern Victoria, and they have everything's color coded. So even the people were color coding that, they had different colored overalls. So the people that were working in close really closely to the baby formula, they actually had to wear red overalls. The people who are working in the raw milk area, they had to wear blue overalls. And then all the brushes, all the equipment was also color-coded. Even the vacuum cleaners were all color-coded. So it was clear as to what belonged in each area, and there was no possibility of contaminating the raw milk. Raw milk might contain something like salmonella to the baby formula. It was obviously a very high-risk product. But you've got similar practices in many restaurants where they've got color-coded chopping boards. Often for the finished product, it might be white or green chopping board. And then for raw products, it's generally a red chopping board and red knives as well. So this is what part B was about, is having a look at how cross-contamination actually occurs. And again, we're going to use the glow germ lotion, put that on a bit of chicken, and then chop up the chicken. You'll see that the chopping board gets all contaminated, the knife gets all contaminated, and then when we chop up the salad, that then also becomes contaminated with bacteria. I found something similar on this particular YouTube video, and when you go have a look at it, you'll see that they've done something very similar. So they've got the lotion, they put it into the chicken, and then they go about cutting up the chicken. You can see all of this contamination on the chopping board. There's also contamination on the knife, and also on the hands of the guy that was actually doing all the chopping up. And so that can quite easily then contaminate the rest of the kitchen. And they showed that it actually contaminates the bench and so on. So yes, I mean, anytime you've got any raw food, so whether it's raw chicken, raw meat, raw fish, even raw vegetables, they are a source of contamination. So they will contain bacteria. And if you're not careful, you'll end up cross-contaminating from raw product to finished product. So that over here where it says GMP, that's good manufacturing practice. So as long as you're following good practice and have separation of the cooked and the raw, you'll minimize this type of cross-contamination. So cross-contamination, we're saying with ending up with bacteria, contaminating bacteria into the finished product. So that was the first demonstration. The second one was actually looking at your sponge or wipes. So these things actually become quite heavily contaminated. Again, a couple of very short YouTube videos, this one in particular, and they did a number of tests and they showed that the kitchen sponge had more bacteria than you could actually find in the toilet. Because the sponges are constantly moist, you use it in the kitchen, it picks up nutrients in the kitchen, and so things start growing in the sponge. So they actually recommend that you replace the sponge every week, or the other thing you probably do is just pop it into the microwave for about 30 seconds, and then that will actually kill off any bacteria that might be in the sponge. So the demonstration would have shown that a dirty sponge will actually contaminate the workbench and other areas as you're actually wiping. So you think that you're wiping things clean, but what you're actually doing, if you've got a dirty sponge, you're just spreading bacteria across from one area to another. And the third demonstration was I was going to show you what happens if you don't wear the gloves correctly in terms of also causing cross-contamination. There's a very good video online with this nurse. She's showing, although she was actually wearing gloves, you can actually still see on the photo here, she did end up contaminating her hands, contaminating her face and other areas as well. So you can't really rely on gloves to prevent contamination unless you use them correctly. Again, when you get a chance, please watch the video. So now we can move on to what we do with our discussion. So with the discussion, well, it's really up to you what you thought, what you expected to happen, and what we actually observed. So in terms of what we actually observed, these are the results that we're going to be using as per these short YouTube videos. So everybody tells us to wash your hands for a full 20 seconds. So hopefully you would expect that if you didn't wash your hands for a full 20 seconds, you'd still have bacteria there. All right, questions over here. Is water temperature important? I don't, hang on. The video didn't actually, sorry, somebody's asking a question. Go ahead.
I was just going to answer question 1. Well,
Yeah.
not really answer it, but with the video, you said that it didn't show hot water. So would we just say, like, with the results that we obtained, it only showed cold water, not hot water, so we don't know if hot water has any.
Yeah. I've actually crossed out the wrong one. I should have crossed out question 1 and left question 2. Because treatment 1, if I go back to treatment 1, so there's treatment 1 showing they're using water with no soap, and then you can compare that result versus the other results where they have actually used soap. So I'll need to fix that up. So I meant to actually cross out question 1, although you can answer it anyway. Is water temperature important? Well, the bacteria have got a membrane. They've got a cell membrane. So when you actually use warm water, what you're doing is helping to dislodge the bacteria off surfaces, including off your hands. If the water's cold, it won't work as well as warm water. So the warmer the water, the more likely you are to actually remove the bacteria. It just helps loosen up the bacterial cell membrane, and they get washed away. Is soap important? Question 2. I think it quite clearly shows that soap is important, because we can see. So when we had no soap and we just did a rinse, we got 0 reduction. And as soon as we start using soap, well, it's not just the soap, it's also the scrubbing. But soap and scrubbing for 5 seconds, you're starting to get reduction. And the longer you use the soap and the longer you scrub, the more reduction you're getting. So using soap is very important. So please remove that crossing out. You should be able to answer that question. Is time important? Again, if you look at your results, I think you can quite clearly see the difference between 5 seconds, 10 seconds, and 20 seconds. So there's 5 seconds. Still some reduction, but still lots of lotion, so lots of bacteria. A bit better after 10 seconds of hand washing. We got it down to 50% reduction. And then when we do a full 20 seconds, you've removed almost all of the lotion, or if you like, almost all of the bacteria. 95% reduction. What's cross-contamination? Well, that's when you're introducing bacteria from one area to another. And that happens when we, for example, dealing with raw foods like raw chicken or raw meat or raw vegetables, or whether a person's hands is moving bacteria from one place to another. So it's the unwanted introduction of bacteria from one place to another is what they're talking about when they're talking about cross-contamination. Does wiping get rid of germs? Well, not if the sponge is dirty. So if the sponge is dirty, all you're really doing is spreading the germs around. I mean, if I'm using an antibacterial wipe, wiping with that would probably help kill bacteria. But if I'm just using a kitchen sponge, which is already containing bacteria, all I'm doing is just spreading the bacteria around. And how can good manufacturing practice prevent cross-contamination? Well, if I follow the right procedures, so in terms of correct procedures would be, make sure I've got separate chopping boards, separate knives, color coding, so I don't use the same equipment for raw foods as I would for finished products, such as salads. Make sure that I'm using a fresh sponge, so I'm not constantly contaminating with a dirty sponge. Make sure I'm using any protective gloves, make sure I'm wearing those correctly and disposing of those correctly. So you can get some answers once you have a quick look at the video. All right, so you've got a set of results that I'd like you to use for both part A, the hand washing part, and also for part B, the cross-contamination part. So you got your final results here, and using those final results, you can then answer all the questions and have a bit of a discussion as well. So hopefully it all makes sense. Any questions about any of that? Okay, that's good. All right, so if there's no further questions, I'll wrap it up. I have made a quick recording of this, so I'll put it up. Well, yeah, I'll put it up on Canvas, and I'll fix up that error that I've made, and fix that up on Canvas as well. So I think I'll actually end up leaving both questions in. All right, thanks everyone. Hopefully see you all again on Thursday.